Good morning, everyone. First of all, I would like to thank the organizers for inviting me to talk today in Enoforum, Italia. So today I'm going to talk about the generation of non-genetically modified yeasts to improve wine aroma and quality. In particular, about yeasts that impart very intense floral aromas in wine. So to begin with, I'm going to talk briefly about the organization I've been working for the last 16 years as a researcher, the Australian Wine Research Institute, or AWRI. So the AWRI is located in the city of Adelaide, which is the capital of the state of South Australia, in red in this map. And Adelaide has a population of about 1.5 million people, while in the whole state of South Australia, about 2 million people live. And the state is about 3 times as big as Italy. And even though the distances are quite big here in Australia, luckily the AWRI is only 1 hour by car from some of the most important wine regions in Australia, such as the Barossa Valley in the north, the Adelaide Hills, and McLaren Vale in the south. And I would like to highlight that the state of South Australia also concentrates about 50% of the total wine production of the country. And in the 2022 vintage, Australia was the 6th largest wine producing country in the world, with about 12 million of hectoliters of wine produced. And here you can see a picture of the building where the AWRI is located in the Wade campus, about 6 kilometers from the city center. And even though this building was opened in 2008, the organization as such has more than 60 years now, and it was established in 1955. Currently, about 100 people more or less work in the Australian Wine Research Institute. And the institute is funded by an organization known as Wine Australia, with matching funds from the Australian government. So personally, and for more than 15 years now, I've been working in the development of wine strains to improve wine quality. And as you know, Saccharomyces cerevisiae, also universally known as the wine yeast, is widely preferred for initiating alcoholic fermentations. And its primary role is to catalyze the conversion of grape sugars into ethanol and CO2. And during this process, a range of secondary metabolites are also produced by the yeast, such as fusel alcohols, esters, and volatile sulfur compounds. And even though these metabolites are produced in relatively small amounts, they are very important for the aroma and flavor of wine. And I would like to highlight that for 60 years now, the AWRI has been isolating, selecting, and developing yeast strains with the aim to provide winemakers with options to differentiate their wine styles while conducting risk-free fermentations. So where do all these yeast starter cultures come from? Well, the traditional and still the most widely used method consists in the isolation of the dominant strain from freshly fermented wine, typically one strain that has produced a wine with outstanding sensory characteristics. And today, the AWRI has a collection of more than 2,000 of these natural isolates. And several of these strains are being used currently successfully by Australian winemakers. However, relying in the isolation of these naturally occurring strains lacks the power and the potential of developing new strains by improving on what is available in nature. And this is very important since there is a growing demand by winemakers for wine strains possessing a wide range of optimized, improved, or novel enological properties that can add value to the wine. So how can we achieve some of these improvements? So, well, over the last 3 years or so, a range of genetic approaches have been used for the domestication of wine strains for industrial purposes, which can be called as the second wave of domestication. And these approaches are similar to those that are applied in the agriculture sector for crop and livestock development. So two of these methods are known as hybridization and mutagenesis. And it is very important to bear in mind that any strains that are developed using these approaches are considered as non-genetically modified organisms or non-GMOs. And today I'm going to focus exclusively in mutagenesis. Well, mutagenesis is a process by which the genetic information, the DNA of an organism, is changed, which results in a mutation. A mutation is a natural process, and it is the driving force of evolution. And one of the possible consequences of mutations in the DNA is a change in the phenotype of an organism, which refers to an observable trait of the organism. And a relevant example in winemaking would be the generation of the Pinot Gris Grigio variety from Pinot Noir grapes. So it is believed that the color difference between these two strains, so these two grapes, is derived from a mutation that occurred in Pinot Noir centuries ago. So as you have seen in the previous example, mutations arise spontaneously in nature, but at a very low frequency. This is why in the laboratory, we have to use mutagens to speed up the normal rate of mutation. And here you can see an example of a physical mutagenic agent, such as UV light, and EMS would be an example of a chemical mutagenic agent. It is very important also to take into account that the dose of mutagen delivery is critical. Too much will be lethal, and most of the population targeted will die. But most importantly, survivors will carry multiple mutations, increasing the chances that some industrially important traits will be negatively impacted. On the other hand, suboptimal doses of the mutagen will create less diversity than is desirable to generate the traits being sought. So this is why it's very important to find the optimal dose of mutagen, which will depend on the yeast strain and the conditions being used. So generating diversity through mutagenesis is usually regarded as a straightforward process. But after that, we are confronted by millions of mutagenized cells that they all look the same. So how do we find the strains with the desired phenotype? Well, the most efficient way of doing this is through selection. But for this to happen, the strains with the desired phenotype, they must possess a selective advantage over the rest of the population. So when a selective pressure is applied to the population, only the cells with the desired phenotype will grow or appear as colonies, while the rest of the population will die. So now in the next slides, I am going to provide a couple of examples on how we have used mutagenesis and selection to generate non-genetically modified yeast with improved winemaking properties. So first of all, I'm going to talk briefly about the generation of low hydrogen sulfide producing wine strains. And later, I'm going to talk in more detail about the generation of yeast that impart intensive floral aromas in wine. So regarding the first example, the generation of low hydrogen sulfide producing strains, the appearance of unpleasant volatile sulfur compounds in wine, such as H2S, is a frequently encountered problem in the wine industry. H2S imparts an undesirable rotten egg aroma, which is commonly associated with reductive notes in wine. And it is also well known that wine yeast is responsible for the production of this unpleasant compound. So a few years ago, in a project that was developed between AB Biotek and the AWRI, a range of non-genetically modified yeasts that produce very low amounts of H2S were generated. So how did we achieve this? Well, a population of the commercial wine strain PDM was subjected to chemical mutagenesis And after this process, the cells were plated in a solid media known as big agar. So in this media, the color of the colony correlates quite well with the amount of H2S being produced by the yeast. So the darker the colony, the more H2S is produced. So after inspecting visually about 16,000 colonies, we were able to isolate a few strains that had lighter phenotype or color than the control strain, PDM. Then the low H2S producing phenotype of one of these selected strains was validated during fermentation in a Riesling juice. So as you can see in the figure, the parent or the control PDM strain, PDM, produced a considerable amount of H2S during fermentation, while the low H2S strain was found to produce 90% less H2S than the control strain. So this strain has been commercialized by AB Biotek with the name of Mauribin Platinum. And platinum is mainly used for the production of white wines and also in sparkling wine fermentation. And these strains represents a tool for winemakers to reduce the production of H2S in wine and enhance varietal aromas and flavors. Okay, in the second example, now I'm going to describe how we use mutagenesis and selection to enhance positive aromas in wine. So in this case, our targets were the fermentation-derived products 2-phenylethanol and its acetate ester, 2-phenylethyl acetate, which impart a desirable floral rose-like aroma in wine. So we targeted these compounds since today consumers, they show a clear tendency towards fruity and fresh wines. So as you can see in the figure, 2-phenylethanol, 2-PE, is mainly derived from the catabolism of the precursor amino acid phenylalanine through the Ehrlich pathway. And once 2-phenylethanol is produced by the yeast, then it can also be acetylated by the yeast to form the acetate ester 2-phenylethyl acetate or 2-PEA, which also has a similar rose and floral aroma. Okay, so I've just told you that these compounds are associated with these rose and floral smells. But the important question is, are the concentrations high enough so they contribute to these floral characters in wine? Well, to answer this question, this graph here shows the odor activity value, or OAV, of both rose aroma compounds in different varieties of white and red wines. So if the OIV is higher than 1, this indicates a possible contribution of the chemical compound to wine aroma. So what we found is that the OIV for the acetate ester in the majority of the white and red wines tested was below 1. So in other words, its concentration is not high enough to be detected by the human nose. In the case of 2-phenylethanol, its OAV was around 1 in average in the white wines and well above 1 for the red wines. So what this graph is telling us is that if we were to develop a wine yeast strain that produces higher amounts of these rose aroma compounds, particularly in white wines, this could have a profound impact in the aroma of the resulting wines. So how can we achieve this? Well, previous research has shown that the production of the alcohol 2-phenylethanol depends on the levels of the precursor amino acid phenylalanine. And we also know that strains resistant to toxic analogs of phenylalanine, they tend to accumulate this amino acid, which results in the overproduction of the alcohol 2-phenylethanol and ultimately leads to intense floral aromas in wines. So taking into account all this information, the commercial wine strain 796 was subjected to chemical mutagenesis. And then after the treatment, the mutagenized population was plated in a solid media containing a toxic analog of the amino acid phenylalanine. In parallel, we also plated in the same media untreated or non-mutagenized cells with the intention to isolate spontaneous mutants. So after a few days of incubation in this media, we were able to isolate about 73 colonies with a higher proportion of mutagenized colonies. Then the selected strains were used to ferment a small volume of a synthetic mast. And 25 of the 73 strains, they displayed a very strong rose or floral aroma at the end of fermentation. So then these strains were used, the 25 strains were used to ferment a small volume of a Chardonnay grape mast. And the production of both rose aroma compounds, 2-phenylethanol and 2-phenylethyl acetate, was evaluated at the end of fermentation. So this graph here, the results of this analysis are summarized in this graph. So in this figure, the results are expressed as the fold increase in the concentration of both rose aroma compounds in the resistant strains and in comparison with the control, which will have a value of 1. So the resistant strains show, in average, a 13-fold increase in the levels of the acetate ester when compared to the control, while the increase in the levels of 2-phenylethanol were a bit lower, about 6-fold in comparison with the control. So taking into account all these results, initially we selected one of these strains, which from now on I'm going to call Rose 1. So this strain was selected since it produced high levels of both rose aroma compounds and also showed really good fermentation kinetics. Okay, so then Rose 1 was used to ferment 20 liters of a Chardonnay grape mast in stainless steel kegs, and its performance was compared to that of the control strain 796. So these fermentations were conducted in triplicate and at 15 degrees. And this is the first of the 3 case studies that I'm going to present to you today. So this graph here shows the fermentation kinetics of both of the wines produced by both strains in Chardonnay. So as you can see in the graph, the rose strain in blue fermented at the same rate, if not quicker, than the control strain, which is depicted in green. And ferments were dry after about 23 days at 15 degrees. Also, at the end of fermentation, a range of fermentation-derived products were also analyzed in these wines. And as you can see here in the graph, the parent strain 796 produced levels of the acetic ester below its other threshold, while fermentation with the Rose 1 strain resulted in a 37-fold increase in the levels of this compound, which is well above its other threshold. In addition, this Rose strain also produced 7 times more 2-phenylethanol than the control. Now, regarding other fermentation products, this Rose strain also produced significantly higher amounts of several acetate esters that are associated with fruity characters, such as 2 and 3-methylbutyl acetate. And also, importantly, the levels of ethyl acetate, which is described as a nail polish remover, were unchanged in the wines made with the Rose strain. So we also monitor the evolution of the rose aroma compounds at different time points. So we at the end of fermentation, then about 6 months after the wines were bottled, and just before the sensory analysis on the wine, about 12 months in bottle. So as you can see in the graph, the levels of the alcohol 2-phenylethanol remain unchanged over time in both wines fermented with the Rose strain or the control. So it is a completely different story for the acetic ester. And even though we observe a sharp decrease in the levels of this compound with time, in the wines fermented with the rose strain, you can see that the concentration of the acetic ester is still well above its odor threshold, even after 1 year in bottle. Also, then a formal sensory analysis was carried out in the wines with a panel of 10 expert tasters. And the results of this sensory analysis are summarized in this graph, which is known as a radar or spider plot. So as expected, the floral aroma was the attribute most affected by the rose strain, increasing significantly, while no significant differences were observed in the other aroma attributes, which are shown in red. In the palate, even though the wines fermented with this rose strain were scored slightly higher in astringency and bitterness, these wines were not perceived in a negative way by the panel of expert tasters. Okay, now in the second case study that I'm going to present today, which was also conducted in Chardonnay, we wanted to assess the effect of different levels of 2-phenylethanol produced by the yeast in the sensory properties of Chardonnay wines. So then for this experiment, we selected 3 different strains. So strain Rose 1, which produces extremely high levels of 2-phenylethanol. Then Rose 2, which also produces high levels of this floral aroma compound. And finally, Rose 3, which produces more moderate amounts of 2-phenylethanol. And as in the previous case study, also in Chardonnay, after the alcoholic fermentation, these wines were bottled, and then a formal sensory analysis was carried out on the wines with a panel of 10 expert tasters. And the results of this the summary of this sensory analysis are summarized in this graph. So as you can see, wines produced with all 3 rose strains, which are depicted in red, orange, and blue, were more highly rated in the rose floral aroma attribute than those wines that were fermented with a control strain. Also, in this graph here on the right, you can see that the intensity of this floral rose aroma attribute seems to correlate quite well with the amount of 2-phenylethanol produced by each strain. And the higher the 2-phenylethanol levels, also the higher the intensity of the floral aromas in the wine. So now, for the same wines, if we focus in a negative aroma attribute in wine, such as cooked potato vegetable aroma, which is often associated with reductive notes, we can see that the intensity of this unpleasant aroma is much lower in the 2 wines that were fermented with the 2 rose strains that produce the highest levels of 2-phenylethanol, which are shown here in orange and blue. Interestingly, when these wines were analyzed for the presence of different volatile sulfur compounds associated with reductive aromas, we found that the concentrations of both methenethiol and methional were lower in the wines that were fermented with these 2 really intense 2-phenylethanol producing strains, as you can see here in orange and blue, and in comparison with the control in green. So this analysis, this might explain why these strains produce wines with less intense reductive notes. Alternatively, this could also be explained as the overproduction of rose aromas by these strains. This might help mask somehow the presence of certain negative aromas. Okay, so now in the last case study I am presenting today, we also wanted to explore what was the effect of the overproduction of 2-phenylethanol in the aromatic profile of red wines. So to assess this, we fermented 50 kilograms of Shiraz grapes with both a control strain and the Rose 1 strain. And the results of the formal sensory analysis on the wines is summarized in this graph. So similarly to what we have observed before in Chardonnay, Shiraz wines made with this Rose strain in blue in the figure, they were rated higher in both floral and honey aromas than the control strain. However, other aroma attributes which are typically seen in Shiraz were masked, such as overall fruit and red and dark fruit aromas. So as a consequence, the wines made with the Rose strain, they were considered unbalanced on the palate and did not meet the expectation for the flavor profile of a Shiraz wine. Okay, so this graph here summarizes the overproduction of 2-phenylethanol by different Rose strains and also in different grape varieties in 5 different vintages. So as you can see in the figure, in all the varieties that we have tested so far, we always observe a sharp increase in the levels of 2-phenylethanol produced by these Rose strains when compared to a control strain. And interestingly, the effect of these Rose strains on 2-phenylethanol production, it seems to be more important in both rosé and red wines than in white wines. Also, thanks to the detailed sensory analysis that we have conducted on some of the wines produced with these Rose strains, now we have a clear understanding of which wine styles will benefit the most from these rose and floral aromas. So these strains, they seem to be highly suitable for the production of white wines, as we have assessed in Riesling, Chardonnay, and Pinot Gris Grigio. These strains also seem to be to perform quite well also in the production of light rosé styles. But on the other hand, they seem to be less appropriate for red winemaking. Finally, regarding the production of sparkling wines using the traditional method, one of these Rose strains has also shown its potential to produce distinctive base wines with desirable floral aromas for fresh and fruity sparkling styles. So now to finalize this presentation, I would like to talk briefly about the genetic characterization of some of the non-genetically modified yeast that we have isolated in this study. So we have sequenced the genome of several of the most interesting 2-phenylethanol overproducing strains to try to find which is the mutation in its genome that will be responsible for this phenotype. So in the 7 strains that we have sequenced so far, we have found mutations in 2 genes that are involved in the phenylalanine biosynthetic pathway, which is the precursor of 2-phenylethanol. And these genes are known as tyrosine 1 and ARO4. Then to demonstrate whether mutations in these genes, in ARO4 or tyrosine 1, are responsible for the overproduction of 2-phenylethanol, we introduce these mutations in a haploid wine strain using a genome editing technique known as CRISPR-Cas9. Then these modified strains were used to ferment a small volume of a synthetic mast. And at the end of fermentation, the levels of 2-phenylethanol were analyzed and were compared to those of a control strain. Okay, so all 3 ARO4 mutations, which are shown in green in this figure, behave similarly and resulted in a 20-fold increase in the levels of 2-phenylethanol when compared to the control strain. The mutations in tyrosine 1 in orange in the figure also behave similarly and show an even greater impact in the production of 2-phenylethanol and resulted in a 40-fold increase in the levels of this compound. So these results show that these mutations are responsible for the overproduction of 2-phenylethanol. So now to summarize this presentation, so I would like to highlight that We have isolated a number of non-genetically modified strains that produce significantly higher quantities of floral aroma compounds during fermentation. We have also shown that these intense floral aromas are well suited for white and rosé wine production. And finally, we believe that these strains are a tool for winemakers to increase sensory quality of wine and potentially the general appeal to consumers. Thank you very much for your attention.
Thank you very much, Toni. Stay with us, please, for the question at the end of the module.
Sure.
I thank Toni very much for his brilliant contribution and for the work performed at the Australian Research Institute, which is an historic partner for us. You see, I would like to briefly tell you now what AB Biotek is, or Biotech is. AB Biotek is a commercial division of AB Mauri, which is a company with a long-standing tradition. I hope you can hear me. Can you hear me? Yes, we can. So this is a company working in the field of fermentation and yeasts. AB Mauri also includes a Mauri Vin assortment for wine. AB Mauri has baking as its core business, which is the reason why we decided we would distinguish our commercial activities in that we do other things than baking. AB Mauri is present all over the world with 52 plants, 32 countries. AB Biotek specifically works in the field of enology, liquors, beer, bioethanol, human nutrition and animal nutrition. And in this session, I will talk about wine. I'm also partly in charge of liquors and also beer. In the past, also animal nutrition. We have dedicated research centers and dedicated production facilities all over the world, and specifically these in Australia, the Netherlands, the US, and Spain, as is the case for human nutrition. At present, AB Mauri products, which briefly so in the presentation, are distributed by Esperti in Italy, and this goes for a Mauri Vin as a brand, but also for Pinnacle, which we are introducing worldwide. Now, rapidly, the floor goes to Stefano Santoro for his contribution, so that he can give us a practical example of applications in Italy, and I'm referring to the application of the AWRI Rosa yeast. Good morning. Thank you for this opportunity to take the floor. We will be extremely rapid and fast in the presentation, because I have a very hands-on approach in my presentation. I would like briefly to reiterate what Toni Cordente talked about, which is the fact that one of the most splendid metabolic pathways is the Ehrlich pathway, whereby the catabolism of the amino acid is entailed and involved, and that is used by yeast to produce higher alcohol, which reacts with acetate and the corresponding ester. In the AWRI rosa yeast strain, this metabolic activity, which is linked to the genomic part of the characterization of genes, has the capability of producing 2-phenylethanol and 2-phenylethanol acetate with this specific and distinguishable scent of rose. The company decided to do the following. We thought we would exploit the natural capability of the yeast to produce this scent and enhance that activity. The yeast is very important in our assortment, so we thought we would further exploit its features. We identified a specific organic nutrient whose profile, whose amino acidic profile, permits to further enhance the production of the yeast of the scent. Here we have 1 and 1 making 3 and not 2. If we now go back to the experience made in Italy, you see here a map where I put as much as possible in the space I had at my disposal, but actually there could be additional names to put there. I decided I would include as many experiences as possible, starting with Friuli, with Pinot Grigio, Merlot, Rosato, then base for sparkling wines, Prix de Mousse, then in Veneto, Valpolicella, Spumanti. That was for Veneto, not for Friuli Venezia Giulia, and that is reds and rosé wines. Luckily, we not only have only Syrah or Merlot or Cabernet in Italy, we have so many indigenous varieties, more than in Australia, certainly, and we have had a wonderful result with those varieties as well. And still in Veneto, Garganega Chardonnay, Tokai Rosso. And we then also performed experiments with the consortium for Chiaretto and Bardolino wines with very good results. Then Trebbiano, going down south, La Crema di Morro d'Alba, Sangiovese, Verdicchio, then Rosati a Sangiovese, Merlot, Vermentino, Vernaccia, San Gimignano, Rosati a Spumanti a Montepulciano, Trebbiano d'Abruzzo, Pecorino, Merlot, in Puglia, Rosati from Primitivo, Negro Amaro and Bombino, Minutolo, Verdeca, Chardonnay, Malvasia, Basilicata. This is the name of the region. Rosato rosé wine from Aglianico, and then we have Gagliopo in Calabria. We had Sicily, you saw the names, and then Campania, Falanghina, Fiano, and so I don't want to confuse you with all the names of the varieties, but you see all the names for the various regions. And now we are moving further north all the way up to Piedmont with Moscato, Bracchetto, Nebbiolo. But if we go back down to Tuscany, again we have Vermentino, Vernaccia, Sangiovese, Merlot. And these are terpenic varieties which feature the rose aromas, which means that we benefit from this further enhancement by working on the yeast. So I understand that it was very fast, but thank you so much for your attention, and we look forward to welcoming you at our stand, where we have a beautiful variety of wines that you can taste, and the glass will talk for us. So as Stefano said,